thanks to the organizers for inviting me, and good morning. So, yes, so this talk will be about the nonlinear dynamics of gene networks, or in other words, of the circuitry that is underlying most biological functions. And probably some questions that have been raised by a few speakers before here is just a few remarks about the fact that living cells must carry out very complex tasks because he has to maintain itself into proper operating conditions. He has to respond to outside signal. He has to find nutrients and to process these energy sources and so on and so on. And then there are very, very different types of signals, which can be physical, biological, mechanical. And then, so cells must carry out complex signal processing and achieve sophisticated decision making. And these processes might be very robust. So the question is, how does this work? So as a very classical example of a complex task, you have cell division. So a basic process to life is the fact that cells can divide from one cell to two cells, and in this process they must grow, they must duplicate DNA, and then break nuclear envelope, and so on and so on. And this has to be coordinated, done in a very sequential and very robust way. And again, what is the logic behind this process? Okay, so another question where gene networks are relevant is, of course, in our bodies we have very different cells. These cells have very different functions, morphologies, from neurons to bone cells to blood cells, and then whereas they share the same DNA sequence. So how can we explain that these cells are different yet have the same genetic material? And then the answer is that the fact that the DNA is not a static object, but carries a set of genes which carry genetic information. But this genetic information is transcribed in a biochemical process which is called transcription. And then so the sequence coding for a given protein is copied into a messenger RNA. And then later on, this RNA is copied into a protein. And then the more important fact here is that the frequency at which the genetic sequence of a gene is copied is not constant in time, but can be at some time very low, at other times very high. In other words, each gene has a certain activity which can vary with time. So, in a sense, at a basic level, as it's suitable for here, we can say that the cell in which the state in which a cell is will be determined and governed by the activities of genes, so the frequency at which they are transcribed, and by the abundances of the different protein species. And so the key quantities that will determine the cellular state are the transcription rate, so at which frequency do we synthesize messenger RNA, and together with the RNA half-life time, this will determine the mRNA level. And then the mRNA level will determine protein translation rate, which is the synthesis rate of proteins, and then which combined with a half-life time of proteins will determine protein level. And then these kinetic rates are not constant. They can be modulated under the influence of some other actors. And then, again, the interesting question here is what determines gene activity. And then what we'll assume here at the basic level is that the activity of a gene, so the RNA synthesis rate, is governed by the fact that there are some proteins called transcription factors which can bind to DNA just before the coding sequence in the regulatory region called the promoter. And because this transcription factor, by binding to the DNA, slightly bend or deform the DNA, they change the frequency at which the RNA polymerase which copies the gene binds to the DNA, and so that determine gene activity. And then you can have activator protein, which will greatly enhance gene activity, and then repressor proteins, and so on. And there can be a very complex cascade of processes because this transcription factor can act alone, or they can bind together to bind to DNA. So they can be active or inactive, modified by other proteins. And then you can imagine a whole sequence of processes eventually determining the gene activity. And of course, now that you realize that the protein which controls the activity of a gene is itself synthesized by another gene, you realize that actually genes determine the activity of other genes. And then, of course, it turns out that if you follow all these links, then you will find that you have very complex sets of feedback loops. And then that eventually, through this feedback loop, so you see here as an example, the protein 1 synthesized by gene 1 governs the transcription of gene 2. The protein 2 controlled by gene 2 binds to protein 1 just to control the transcription activity of gene 3, and so on, which means that eventually the activity of gene 1 is controlled by this complex feedback loop, and that eventually the dynamics and the variations in the state of gene 1 eventually come back to itself just to modify itself. And then, of course, as we will see, even with very simple feedback loops, you can already have a complicated dynamics. And then there is a whole field of study is just to understand which kind of dynamical phenomena you can observe in such systems. And the idea is that even with mere intuition, it's not possible just to imagine the dynamical processes that you can observe. Just to get a very vague feeling of the complexity of cellular networks, here are only a very limited set of actors which have very important roles in the cellular machinery. For example, here you have all the molecular actors of the cell division cycle, the cyclins, CDC25, and so on. The NF-kappa B system, which is involved in the inflammatory response. P53 and so on, which involve in the response to DNA damage. And then you see that not only you have some subsystems which are very strongly coupled together and which can have really complicated dynamics, but also all these systems are coupled together. And then the question is what can we do just to understand already at a qualitative level these dynamics. Okay, and then here what I will focus on mainly is just to the fact to view some gene networks or regulatory networks as a dynamical systems. And here you have a few examples of small networks on which display complicated dynamics and which have very much studied in the last years. So again, the DNA damage response system with P53 MTM2. And here is a system that shows excitable behavior in Bacillus subtilis and which triggers a special state, cellular state called competence. And again, NF-kappa B. And all these systems, I mean, just for example, the P53 system and the NF-kappa B system respond to external stimuli, DNA damage or pulses of tumor necrosis alpha. But then they have also their own dynamics. And just the goal of so it's why it's important just to study the dynamics of these networks. And then, of course, if you talk about a dynamical system, you must define what the state space is. And here we will mainly consider that the variables are the protein and RNA concentrations for each of the species involved in the network, and possibly also gene activities. And then, of course, we'll see that through all these links in these networks, the time evolution of each variable depends on all the other variables, directly or indirectly, which means that you have really a global and collective dynamics.
Some of these links act on other links, and some of these links act on proteins. So how should we understand this?
Okay, that's a good point. It's right, because you mean like here. So here means that it's not a direct modulation of the actor, but it is a modulation of the way this actor acts on this actor. For example, if you, it could be that, I mean, a direct link could be the fact that some protein just activates the transcription of another protein, or this kind of link could be the fact that the protein modulates, for example, the binding of the other protein to DNA. It's a way to say that the protein, one protein modifies the properties of another protein instead of just activating its synthesis or its degradation. Okay, and then, of course, in these examples, okay, so here, for example, okay, this is a good example. So make A just stimulates the degradation of COM-K, but when COM-S is present, COM-S binds to make A and then prevents make A from triggering the degradation of COM-K. Okay, so here we see right, so even small networks, but with a lot of links and feedback loops. And then it turns out that even most of the talk will be done with very simple feedback loops featuring 1 or 2 proteins. And it will turn out that there will be a very important distinction. So again, so the simplest feedback loop is when the gene just modulates its own expression. And of course, there is a very important distinction between positive autoregulation and negative autoregulation. So positive autoregulation, it means that the protein product of gen X here comes back and binds to its promoter to activate transcription. So it means that the more X is active, the more it will be active. And then the other important class of self-feedback is negative autoregulation, where the protein product, for example, here of gene Y, comes back and represses transcription. And then it turns out that you have very different dynamical behavior between these two classes, and this is what I will try to detail. Okay, and just to show that it's not always easy just to understand how this will behave, here you see that you have 3 time courses. So you have a gen X here, which is activated by a signal Y. And then one of these time courses corresponds to the unregulated X, one corresponds to the self-repressing gen X, and one corresponds to the self-activating gen X. And then the question is, among these 3 time courses, which is the one corresponding to self-repression and which is the one corresponding to self-activation? So, okay, so okay, so for example, let's take the red one. Does it correspond to self-repression or to self-activation? So those who believe that it's self-activation, please raise your hand. Okay, so there is a minority who is right, actually, because here it is self-activation and here it is self-repression. So of course, because it goes slowly here, you feel it is repressed, and then it's very fast, and then goes very fast to the equilibrium, and you feel that it's activated. But actually, what you see is that the red curve goes faster and faster as the protein level increases, which means that transcription is helped by the presence of the protein, which means that the protein activates the gene. Here, it's very fast, but as soon as protein level is built, then it goes slower and slower, which indicates that the protein just is repressing transcription. And so you see that it's, even with this simple system, it's very easy to make the wrong prediction based on just intuition. And this is why you have to understand the dynamics and the, even the qualitative dynamics of such systems. OK, and then just as a preview of what we will discuss in more detailed way here, it turns out that usually positive feedback, so self-activation, will give us some bistable system, states which can, systems which can have several states. And it's why? It's just because you see in this example, if your gen X is just at 0 activity, it produces no protein, and then it is not activated because there is no protein produced, and then it stays in an inactive state. But if you just bring a certain quantity of the X protein inside the system, then it can activate, and then the gene will maintain its own activation. So basically, usually the positive feedback will be associated with system where there is a self-sustained state, some kind of virtuous circle or auto-reinforcement. And negative feedback will be linked to homeostasis. So negative feedback is you have a fluctuation, and this fluctuation is corrected because the feedback comes back inverted with respect to the initial fluctuations. So usually it's a good way to maintain equilibrium. But as we will see is that in some cases, if this negative feedback is delayed, this is how you can trigger oscillations. And so on. And of course, so you can just try to see different network topologies with different number of vectors, different sets of feedback loops, and just to understand if there are some general rules or some design principles which can help you to understand the dynamics of these networks. All right, and of course, well, this could be modeling and theory, but what was really nice is that for now, for a couple of decades, it had been possible just to monitor the dynamics of molecular events in live cells, just using fluorescent reporters. And then I talked about the p53 MDM2 system. And then it's an example where you have two fluorophores attached to the p53 protein and giving green light, and MDM2 protein giving red light. And in this way, each of these panels here just gives you the time evolution of the p53 and MDM2 concentrations in real time. And then, of course, it is really invaluable because it's when you just try to build mathematical models of gene regulatory networks, then you are claiming that you describe the time evolution of protein concentrations, mRNA concentrations, and then thanks to these experimental techniques, you can really measure what it is and then make detailed comparisons with your models. And of course, this is a fantastic stimulus. Okay, so of course, once you have a network of molecular interactions with kinetic constants, there are different ways in which you can turn it into a dynamical system or a mathematical model. So here, I just take the point of view that We have a large number of molecules so that we can use things like the law of mass action. And then I will mainly turn these complex networks of biochemical reactions into a set of differential equations. And these differential equations basically will describe the time evolution of the concentration or activity of these species. For example, here is it gives a time evolution of one of the messenger RNA in the system. And of course, with this system of differential equations, well, we can do numerical simulations, but as we will see, we can do also analyze which kind of qualitative changes occur in these equations and do bifurcation diagrams. And just for example, in this case, it's a mathematical model for a biological clock, and then just to study when do we observe the appearance of oscillations. Okay, so just a few slides of how exactly you turn these you translate these biochemical processes into ordinary differential equations. So the goal here is not so much to show, to build a detailed equation into specific equations, but to make the point that, of course, if you look at the most basic processes, we will get linear equations. But we will see that as soon as we consider all the possible molecular interactions that actually do take place in cells, then it's very easy to get nonlinear equations, and it's also very easy to tune and to modify and to modulate this nonlinearity as you want. So I guess probably the simplest processes is just the degradation of a protein. So it's kind of similar to radioactive decay. And then over each time interval, you have always the probability of one protein to be degraded is constant. And then this gives a very simple equation like here with an exponentially decaying solution. And then if we want to understand what determines protein level, the other important reaction is the synthesis. So it's a bit, well, from a point of view of conventional chemistry, is a strange reaction because the RNA is both in the reagents in the products, but basically this expresses that the sequence in the RNA is copied into the protein, but this indicates that, again, the synthesis rate of the protein is proportional to the concentration of the RNA. So that's another important equation, because here we describe the synthesis and the degradation of a protein, which means that if we coupled the two processes, then at least we can, assuming first that RNA is constant, then we can describe how protein concentrations evolves over time. And then we see that basically what is important here is that in this case, because it's one-dimensional, there is an equilibrium state here, and if you start from a low protein level, you increase protein to go to this level. If you start from a high protein level, you decrease to that level, because here degradation is more important, here synthesis is more important, and when the two balance, then you have this equilibrium. Well, here it's quite simple. It's linear equation, so really no nonlinear dynamics. But as you know, and perhaps this answers the previous question, even if these simple processes, it's very common that there is a third actor that is involved and just will modify the process. And then, for example, assume that we have a repressor molecule R here, which can bind to the RNA M, and it assumes that when RNA M is in this bound state, then it can no longer be translated. So only the free M is involved in protein synthesis. If you just assume this process is very fast, so that it is always at equilibrium, that the total quantity of bound RNA and free RNA is constant, then it's very easy to find that the free RNA concentration is related to the total RNA concentration by this relation, and then the equation is modified as follows. Then the interesting thing is that if R is itself a dynamical variable, which changes due to other equations, we see that we had a perfectly linear equation, which has suddenly become nonlinear. And what is interesting is that usually when you look at a very complex network, you are always asking what is the purpose of this or this interaction. And then we see that sometimes the interactions help to build the feedback loops that will really be the backbone of the dynamics. And sometimes interactions can be there just to modify the kinetics and just to bring the kind of nonlinearity which may be needed to trigger certain dynamical processes. And then, because there are so many of these, there are so many ways in which these processes can be regulated, you can imagine that basically you could generate any type of nonlinear equation in this way. Another way to generate nonlinearity in system is one process which is very common is dimerization. So you have two proteins which bind together and together make a complex. And it's very common that this complex has properties that the individual proteins did not have. For example, it can be that the dimer formed by these two proteins, only the dimer can bind to DNA and regulate transcription. And then, again, because the complexation rate depends on the square of the monomer concentration, then you have a nonlinear term here. Well, yeah, that's a nonlinear term in the case of homodimerization, but it's still a nonlinear term in the case of heterodimerization. So it's another way in which a nonlinear term can enter the kinetic equations. Even the degradation, which I presented as simple, then it turns out that in most cases, it's not spontaneous degradation. Usually, the degradation of proteins is catalyzed by some enzymes, and then the protein binds to the enzyme, there is a complex, and then the protein is degraded. And then it turns out that this process can saturate, which means that the kinetic, well, I do not enter the details, but the kinetic can be described by this equation. So instead of having a straight line here, you have a curve like this. And the important thing is as degradation saturates, which means that if you have, you can only degrade so many protein per second, and then whatever is above will not be degraded. And as we will see, it's a very important ingredient to generate oscillations. And then it even turns out that if you have more complicated degradation schemes, with more complicated enzymes, you can have really more complicated kinetic laws, such as here. And you see here a phenomenon which is called cooperative stabilization, which means that usually it's normal that the more proteins you have, the more you degrade per unit time. But it turns out that somehow, sometimes perhaps they can bind together and they can protect themselves from degradation, which means that the more proteins you have, actually, the less you degrade. And in this case, you have some kinetic laws as here, where you see that first you have a positive slope and then a negative slope. And again, as we will see, such a negative slope is a key ingredient to generate oscillations. But again, at this stage, the message I want to convey is the fact that there are so many ways in which to organize a certain biological process that you can really have very different kinetic laws. And again, evolution can really tune, just add a process, remove a process, such as to tune exactly the kinetic law that will be needed. Just an interesting fact from the point of view of dynamics is you see that, as I discussed before, when you have normal degradation or spontaneous degradation, the solution is a decaying exponential. So as you know, exponential decay is very fast. But then it turns out that when you have saturated degradation, then it turns out that at some point, when the when you have a decay, it does not decay along an exponential, but along a straight line, because basically the number of proteins degraded per unit time is fixed, and in fact corresponds just to the number of enzymes present. And then in this case, okay, so I think, okay. So in this case, which means that the time it will the time it will take just to go to 0 will in fact be proportional to the maximum amplitude of the signal, which means that you will decay over a much longer time. And it's interesting that in some systems it seems that tuning the degradation so that it becomes saturated is being used just to generate a time delay of a sufficiently long time delay. And okay. A very important, another very important source of nonlinearity is the transcriptional regulation. So in a very simplistic view, you can assume that you have an activator, a regulatory sequence on DNA, and then that's when the activator binds, then you can have transcription. And then you have some kind of equilibrium here between the free and bound DNA. And then if you assume that you have equilibrium, then it's easy to see that if you want to compute the fraction of time f where the regulator is on DNA, then you have this equation where k naught concentration of A times 1 minus f equals k1 times f, because it's just assuming that you have as many transitions in one way as in the other way. And then, of course, you can assume that you have your transcription rate, so the gene activity, the RNA synthesis rate, is just an average between these two cases. So you have R0 transcription rate in the unbound case and R1 in the bound case. And then when you just combine with this relation, you find that the transcription rate has this kind of expression. So again, you see that the gene activity is directly a function of the concentration of the protein that controls it. And you see that, again, this term is nonlinear, so that's another source of nonlinearity. And, of course, you can combine all this. So you assume that the gene is not regulated by a monomer, but by a dimer. And then, again, when you compute the expression, so I do not go into the details, but it's just that you get the idea. When you compute the transcription rate here, you will find that instead of having something like 1 over 1 plus A over some threshold, now it's the square of these ratios which is present. And if you just assume that you have not a dimer by a tetramer, then it will be 4. And so on. And in general, the exponent here that occurs is so important that, I mean, there is a name for it. It's called a Hill coefficient. And this type of expression comes very often in the analysis of cellular dynamical systems. And really, it quantifies how fast you make the transition. So you see here the red curve is for n equal 1. The green is for n equal 2, and so on. And you see that when the Hill coefficient is very low, you have a gradual transition, whereas you can have really a switch-like behavior when you have a high value of n. And now, why do I mention this is because actually there are several ways to have effective Hill coefficients that are very high. For example, one example, sorry. Yeah, but it's just it's just because I have a problem. Okay, see. Because I had the bar, which now it's gone. Okay, so it's a nice example showing how just a simple interaction can really modify your kinetics. Now you have a transcription factor A, which activates the gene O. So usually you would have, and here it is the concentration of protein O as a function of the protein A. So normally you would have this relation. And now you assume that there is a protein B that can titrate A away, so that when A is in the bound state, it is not active and cannot activate the transcription of O. And then it turns out that under some threshold for the concentration of A all A will be bound to B, and then it can absolutely not activate the gene, which means that there will be a very sharp and sudden threshold where suddenly the gene gets activated when finally there are enough A to overcome the complexation and the binding to B. And then in this case, what is observed is here, and you see that you have an effective yield coefficient of about 20, more than 20, which means that you have exactly, sorry? Yeah.
So I don't get exactly what you mean there. So at the threshold, are they separating?
No, it's not that separating, it's that all B are bound to A and cannot bind additional A.
So as, for instance, A increases, why doesn't it start gradually going?
Yeah. So the normal behavior is as you bring additional A's, there is a higher and higher fraction of time during which A is bound to DNA, and so you have a gradual transition. But then when you increase, now when you have in the presence of B, as you increase the concentration of A, all the A you bring just get bound to B and do nothing, and so you observe no effect on transcription, and suddenly there is enough A that all the B are bound and can no longer be part of the game, and then suddenly all the A you bring additionally do stimulate transcription. So it's really, and then the transition is much sharper because you have just to overcome this set of blocking B's. But the point here, so you see here, so you get exactly the same behavior as if you had to build a complex of 20 copies of A. But there, it just occurs with a much simpler process. And then the idea is also just that I want to convey is that if you want to have a very sharp and very nonlinear behavior, it can occur from very, very simple set of processes. And again, a very classical example also is phosphorylation cascades. And then the classical system is the MAPK cascade, in which you have a signal which phosphorylates a kinase. This kinase triggers a sequence of phosphorylation in the stage below. And itself, this kinase here controls phosphorylation here. And what turns out is that at each stage, the relation between the phosphorylation state and the input stimulus is steeper and steeper. And what you see here is the effective yield coefficient. And again, it's another way in which you can really make a process very nonlinear. You just put a phosphorylation cascade somewhere in the pipeline, and then you can really tune the input-output relation as you want. Okay, so I'm done with the first part. So the important ideas here is that, I mean, when you think about cellular biological functions, they are implemented by molecular interaction networks. And the important idea is that these are strongly nonlinear dynamical systems, which consists of many interlocked feedback loops. And even simple loops can have complicated dynamics. And then there are really millions of ways in which you can make things nonlinear by using protein complex formation, cooperativity in transcription regulation, and so on and so on. So the basic idea is that really nature, I mean, just with over times, in times over which evolution can do something, can really tune and modify your dynamical system so that the desired dynamical behavior can be realized. Okay, so now I will try and come back to gene network as a dynamical system, and I will try to get rid of equations and just to see that there can be a way to describe the dynamics in a geometrical way. And again, I'm coming back to this simple system, which is a gene regulated by its own protein. And then the simplest model you can write of this system is this one. So you have two species, RNA. So this is the transcription rate, the RNA synthesis rate, and degradation. And again, and this is the kinetic equation for the protein. So synthesis is proportional to the RNA, and this is the degradation. And again, I would like to think of these equations in a very simple way as expressing a balance. So if we have a dynamics, it means that the protein changes, concentration changes over time. So when it increases, it's because synthesis is stronger than degradation. And when it decreases, it's because degradation is stronger than synthesis. And it's all about a balance between the two. And then it turns out that a very nice way just to get a global picture of the dynamics is just to represent this dynamical system in a state space. So as you see here, so you have the two state variables, protein P and RNA M. And then what do these equations tell? They tell you that for each pair of M and P, you can compute the time derivatives for P and M. And this is a velocity vector. So here you see each arrow in this plane is the velocity vector dp over dt, dm over dt. And then it's very simple because if you want to understand the dynamics of the system, instead of trying to solve the equations and to find an analytical solution, then it's an orientation run. You just here and you follow the arrows and you follow the arrows and you move in this state space. And for example, if you start from here, you see that you just follow this trajectory. And in this case, this trajectory converges to a fixed point, which is here. Okay, so again, so it's very, and because you can have a global picture of all the velocity vectors in this state space, you can have, at a glance, a view of the dynamics. And in this case, you can see that there is a global attracting state. There are two very important curves here, the green curve here and the blue curve. And these are the so-called null lines, which correspond to one of the two derivatives being 0. OK, so the blue line is dp over dt equals 0. And then over the line, it's negative. Under the line, it's positive. So remember what I told before with a simple example with just one equation. Usually when protein is low, it goes up, and then it's high, it goes down. So you can see that above the blue line, all the arrows are going down. And below the blue lines, all the arrows are going up. And now the green line here corresponds to the dm over dt equals 0, which means that left of this line, m increases, and right of these lines, m goes down. And you see that indeed all the arrows below the green line go up. Sorry, so I think I messed. So I'm doing it again. Sorry. So below dp over dt, yes, that's it. So sorry. So just forget what I told. I'm doing it again. So of course, p is horizontal axis. So below dp over dt equals 0, dp over dt is negative. So the arrow goes left. And over the line, all the arrows go to the right because dp over dt is positive. Okay, so left here, right here. And then over the green line, m is decreasing. So arrows are pointing downwards. And below the green line, all the arrows are going up because m is increases. Okay, so the idea is that you have 4 regions. And if each of these 4 regions, you have the same direction. So you see here, P increases, M increases. In this quadrant, P increases, M decreases, and so on. And of course, there is a very special point here, which is intersection of the two. And then, of course, the two derivatives are 0, which means that it's a stationary point. OK, and then you see that here you have a spiral which converges to this point. But then, the idea I want to convey here is that the geometry of these lines and how they intersect really governs the global dynamics of the system, and which makes this picture very attracting because you can really read and understand what is going on. And sometimes just when I will study oscillations, I will make use of this diagram. So of course, it's only practical for two-dimensional systems. Yes?
Sorry, could you go back to the equations, like one slide?
Yes, sure.
So you also have this linear degradation term instead of the exponential term. So
Yes, but
we only see like the first part of the node line, because then the dB/dt node line should also be a Hill function, right? So in the next slide, the blue line seems linear.
Yes. No, it's true. It's perhaps not exactly the same equation. No, you're right. In principle, the blue line should be curved, just to indicate saturation, and that is probable that I made this picture using a set of equations where degradation is linear. Okay. So thank you for. But yeah. And then, it's a very good point, and perhaps we can elaborate because it will be important then after. It's true that the equation for dynamics of P so dP over dt equal beta m minus well, a certain function that I will call f of P, what I will call the degradation function. And dP over dt equals 0 means that we have m equal 1 over beta f of P, which means that really, as you really nicely pointed out, the shape of the null line is really indicating the kinetics of your degradation. So which means that because here it's a straight line, it means that I took a linear degradation term, or a really unsaturated enzymatic degradation. But usually I should have something like this. Okay, so that would indicate saturated degradation. And it's important because by binding this line, it can be an important ingredient for generating oscillations. And, okay. And then, of course, I mean, it's one way to see the trajectory, but of course, you can also represent a time trajectory in the classical way by giving the two concentrations as a function of time. But really, what is nice in this representation is that you can follow the whole trajectory for time going to infinity, and also to see what shapes the dynamics of the system. Okay, and then, of course, so you understand that it will be very important. I mean, just you see that intersections are very important because they correspond to stationary points. And then it turns out that when you modify kinetic parameters, it can happen that the number of these interactions are modified. So you see here you have curves with a S-shaped, and then you tune some parameters, and it turns out that at some point you have only one intersection, and then 3, and then 1 again. And of course, you understand that this will change the dynamics of the system, and that this will be important events. Okay, which means so that usually all the tools from nonlinear dynamics are very important because when you write a gene network system, even the simplest one as here, these equations are very nonlinear. And then you can just study what are the different dynamical regimes observed in different regions of parameter space, and just to understand how this relates to the function you are trying to implement with this circuit. Okay, so now the talk will be divided in two parts. So I talked about positive feedback and bistability, and on one hand, on the other hand about negative feedback and oscillations. And I will try to give a few biological examples of the two behaviors. And then, again, so I'm coming back to this phase portrait description of the dynamical systems, but here it's no longer a self-repressing gene, but it's a self-activating gene. And in the same way as the one null line is related, so you see this null line is related to degradation, and this null line is more or less gives, I mean, if the degradation term was linear, this null line somehow gives a transcription rate as a function of the protein concentration. So here, in the self-activating gene, the blue null line here gives the transcription rate, the gene activity as a function of protein concentration. And so you see that at low protein concentration, the gene is not active, and at high protein concentration, it becomes much more active. And then here I assume linear degradation, and then you see that what is quite interesting is that in this case, you get 3 intersections. And if you look at all the arrows giving the velocity vector in state space, you see that basically you will go to one of these fixed points, or to the others, which means that you have a bistable system. And that really, the point on which you will settle down basically depends on where you started from, so it depends on the history of the system. Okay, so another way to see it, more with the equations. So these are the equations for the self-activated gene. And then if you just look at the steady state equation, then because you can eliminate aim here, it goes down to this simple equation. And then so the green and blue curve correspond to two values of this coefficient here. And the magenta and the red curve correspond to two values of the yield coefficient here. And so again, you see that depending on the value of these parameters, you can have 0 or perhaps 1 or several intersections. So you see that when you consider the magenta curve here with the blue curve, again, you get two different solutions. Okay, so again, this is theory. Does it work? And of course, it's interesting to look at natural, real systems. But just if you argue that you want to understand the dynamics of a simple gene, it's also very interesting to take a synthetic system. And then in this experiment, the authors just engineered a system where you have a self-activating gene. And then they observed that depending on the kinetics and on the cofactors of the process they put in the cell cultures, they can observe bistable behavior or not. So here they have a fluorescence reporter. And in these pictures here, they gave the distribution of fluorescence intensity in the cell cultures. And basically what you see is that in some cases you have only one peak, which basically cells are more or less in the same state with some variability. But for some other values, changing the kinetic constant of this self-activating loop, then you observe two peaks, which correspond to the fact that the cells can be in this state or in the other. Basically, the gene, this gene circuit in the cells can be in the off or in the on state. And this is shown here, whereas in this case you have really all possible shades of green, but whereas in this case it's really dark or green, showing again experimentally the existence of this bistability phenomenon. Another synthetic bistable system, which was published about at the same time. Well, it's basically the same system, except that you have two genes. And then, so when you have two genes, you can have cross-activation or cross-inhibition, and in both cases, it will be a positive feedback loop. So if A activates B and B activates A, the net effect is that A indirectly activates itself, or if you repress your repressor, you also activate yourself. Right. But the important thing is that if you have a fluctuation, it comes back to you amplified. And again, they also observed typical bistable behavior. So you see here they induce the system with IPTG, so just a small molecule forcing the system in the on state. And then when they remove this inducer, you see that the the axis here is correspond to fluorescence intensity, which means gene activity, and you see that the gene stays active. So it's different constructs. In all these cases, you see that you have a typical behavior. And then when they put the cells at 42 degrees, then they remove the bistability, and then it goes down. And again, so again, when you induce, when you use the on state with IPTG, you see that as time goes by, you see progressively all cells leaving the off state and going to the on state. So this is the distribution of fluorescence and so of gene activity. And again, another signature of bistability is how you make the transitions. And then you have two cases here in red and in blue. And in blue, you have a gradual, progressive transition to the on state. But then in the red case, you see that suddenly you make a sudden and sharp jump, which is typical of a bistable system. And we will see other examples. Another example, which was very important also. So there is no gene transcription. It's signaling cascades. And again, I talked about this phosphorylation cascade. So MOS phosphorylates MEC1, which phosphorylates MAPK. And there is a positive feedback by which the MAPK activates MOS. So again, you are in an off state and everything stays off, or if you are sufficiently active here, it maintains itself in the on state. And then it's used in the maturation of eggs. So in Xenopus, it's a frog. And then eggs can stay inactive, I mean just dormant for several weeks. And then when you have a pulse of progesterone, they get into an induced state where they can be fertilized, and then embryogenesis begins. And if you just look at a set of population of ovocytes, if you look at the phosphorylation state of the MAPK protein, then it's very gradually rises. But if you look at the individual ovocytes level, so you see here each column corresponds to a different egg, and then if you have the black bar up, it corresponds to the phosphorylated state, and down it's the non-phosphorylated state. And you see that for each egg, you are either phosphorylated or not, but there is no combination of both. So it's really actually at the population level, it's a graded response. At the individual egg level, it is all or none response. And again, how do you explain the graded response is the fact that as you increase the concentration of progesterone, then the number of eggs which are in the up phosphorylated state increases. And again, it turns out that this system can be understood by the same kind of diagram as before. So again, you see it's a system which is coupled to its input. So this is the red curve is the response of the system as a function of the input. And because the output must be equal to the input, you look at the intersection with the diagonal. And then it turns out that depending on the progesterone concentration, you have this curve or this curve or this curve. Progesterone basically makes the red curve go up. And as the red curve go up, you see that it loses some intersections here. And then for this curve, there is only an intersection in the high state, which means that you can just describe this behavior by the following diagram. So here you have the concentration of progesterone. Here you have the activity of the MOS kinase. And then for a lot of progesterone concentrations, you have 3 possible states, 2 stable, high, and low. And then as you increase progesterone, here you follow, and suddenly at this point, there is only one solution, which is up. And then as progesterone goes down, then you stay on the upper branch, and which means that here you have a cellular memory. The system will remember that it has encountered a progesterone pulse previously in the history. Another example linked to the cell cycle. Okay, so then in this case, so it really works at a cellular memory. Another very important example of bistability is in the cell cycle. So here it's also in Xenopus eggs, and it's sperm nuclei which have been put together with cell extracts. And basically you can see 2 states here. You can see round blue circles. So this corresponds to a case where the nucleus has still its nuclear envelope and where chromatin is decondensed. And here you see that there is no longer a nuclear envelope, and you see that it's very tight. It's because chromatin has been condensed. And here it is the entry to mitosis, so the beginning of the cell division. And so basically here, the nucleus is in a stable state, and in this state, it is ready just to begin cell division. And then the author, what they did, they just increased the amount of, there it's what will, so what has a bistable here is the activity of a certain complex, cyclin B, CDK1. Well, I will not give the detail, but it's the activation of this complex which really triggers cell division or mitosis. And then the author changed the level of a non-degradable cyclin B, and then they see how things evolve. And then they look at time 0 and then 90 minutes later, and you see that to see the transition from a low activity to high activity here, the concentration of cyclin B must be over 36 or something. But now, they start from the high activity state with a condensed chromatin and a broken down nuclear envelope, and they decrease the amount of cyclin B, and you see that now they observe the inverse transition at much lower levels of cyclin B, which basically means you have something like this. You increase cyclin B concentration. At some point, you jump to a high activity state. And when you come back, you have to go significantly lower just to fall down on the lower branch. And then what is the use of bistability? Is commitment. I mean, when you start cell division, you are not going to go back because just you start a lot of processes. It would be very complicated just to unwrap and just to go there. So this system ensures that once you give the signal for division, you just switch to a upper branch, and then you stay there unless you have significant perturbations. And then it's just a way to commit to the cellular decision you have made. And of course, you have also similar systems in the cell cycle, just when, for example, you begin to replicate DNA. So imagine you begin to replicate DNA, and suddenly you decide that it's not the right time, and then you go back. No, I mean, all the machinery is designed so that once you have started the process, you just go to the end. And bistable systems are a very good way just to make this happen. OK. Okay, and then it's interesting because, there are two people who have done a lot on cell cycle modeling. It's John Tyson and Bela Novak. And they had a lot of arguments and debate about what is the use of bistability. And then, of course, you know, a lot of people for a long time have been aware that there is some kind of irreversibility in the mitosis process because, I mean, except in some very, very special conditions, when you start cell division, it never goes back. It's always unidirectional. And for some time, there was a lot of conventional wisdom about the fact that this was due to the fact that just to exit mitosis, you have to degrade cyclin B. And then people were saying that, of course, when you degrade the protein, it's irreversible, so that's the cause of reversibility. But not. Actually, what is really making these cell cycle transitions is really the fact that you organize the dynamics around a bistable cycle, and that when you jump to a remote branch, that you are not coming back very easily. Okay. Perhaps a last example, and then after we will switch to oscillations. It's interesting because it will illustrate another use for bistability. It's about the metabolism of E. coli and the fact that it can use two types of sugars, glucose or lactose. Glucose is preferred, but if you have more lactose and low glucose, then it may be better just to use lactose. And again, you see that this is the entry for glucose, this is the entry for lactose. And then this is a set of genes which are involved into the process and metabolism of lactose. And then you see that when glucose enters, then you block the entry of lactose here. And also, if you follow the links here, you see that more or less you just repress this set of lactose use genes. On the other hand, here you have a double repression. So lactose entering represses LAC-I, which represses this gene. So again, when you repress the repressor, you activate. The enemies of your enemies are your friends. And then, again, there is here a loop by which LAC-Y favors the entry of lactose. So here you have a positive feedback loop. So in principle, in this system, you have a bistable system in which you have two states. One state is using lactose, the other state is using glucose. And then the authors, again, engineered these cells just to report the activity of the lactose system. And then, so here, cells which are in green actually are using lactose, the other are using glucose. And then, so TMG is for the way kind of lactose. What the authors, so you have a distribution of fluorescence, so the distribution of the cells between glucose using state, low activity here, and a high in a lactose using state here, high lactose system activity. And you see that as we increase lactose in the extracellular medium, more and more cells switch to the lactose using system, but basically you have two populations. And then when you come back, you see that even though you decrease the amount of lactose quite a lot, it takes some time before you make the switch again to glucose, and which means that there is, as a function of extracellular glucose and extracellular lactose, there is a gray region here when you can have both behaviors, and it really depends on where you are coming from. And here the use is easy to understand. It's because if you are in a situation where both nutrients are available, you are not going to switch between the two systems just randomly, constantly over time. Once you have committed to one metabolic choice, you stick to it unless really this source of nutrient is really going away. And again, we can see that, again, that's another use for a bistable system. Okay. Okay, so perhaps I'll which switch to oxidation. And perhaps one last word. Also, in cells undergoing apoptosis, so when cells do suicide, where they commit to cellular death, there is also a bistable system. And again, it's because once you have triggered apoptosis, you are not going back. And then, but then here, so it's what is imaged here with a fluorescent reporter is the activity of the BAX gene, which is important for apoptosis. But what is interesting here is that you see that you have significant delays, difference in the delay before switching. And this is really a signature of a critical slowing down when you are near a bifurcation point. Because here, when you have a bistable cycle, if the stress just brings you exactly at the bifurcation point here, everything is very slow and it takes a lot of time to go up because there you are in between the bistable and the monostable region. But when the stress brings you far away from the bifurcation point, then you switch suddenly. And then this corresponds to a state of stress which is very close to the bifurcation point. And this corresponds to much higher stress where you have a much sharper and much quicker transition. And again, well, all these examples are interesting because they are very typical of a bistable system and actually could be observed in quite different systems in optics, in mechanics. These are really signatures of a bistable system. Okay, so so again, a summary for the bistability. Well, so bistability allows us to have several cellular states, and usually it's associated with a positive feedback loop. And then there are many uses just to have a cellular memory. What did I encounter in the past? Just to guarantee that you are committed to critical cell decisions, that you are not going back when it's important to not go back. And then also just to guarantee some kind of permanence and to avoid continuously switching between different alternatives. Okay, so I'm going back to the safe repressed gene. And again, in general, a negative feedback loop is associated with the homeostasis. So you basically, you have some fluctuations, and the negative feedback loop brings this fluctuation down. But what I will try to explain now is that under some conditions, it can also lead to oscillations. And I will try to show why oscillations are important and useful in cell biology. And the first case, the first one key ingredient for generating oscillations is the existence of a time delay. So usually, an example which is very simple to understand because everybody has experienced this is how to regulate the temperature of your shower. So as you know, usually it takes, once you turn the knob, it takes a lot of time for the temperature to adjust. And usually this leads to oscillations in temperature because you just turn the knob and then it's, okay, it takes some time, but then you realize that you turn it much too, and it's too hot, and then you just turn the knob the other way, and then it becomes too cold, and so on. And what is the reason for the oscillations in the temperature shower is just because there is this large delay between your action and your decision and the effect of your decision, and which means that you overshoot the ideal functioning point. And here it's about this, well, this is what is explained here. So just to be simple, the authors here take not a two-dimensional system, but a one-dimensional system. They assume that the RNA dynamics is trivial. And then they just consider that the degradation is instantaneous. So you degrade what the proteins you have now. But the synthesis, this depends on how much protein you had some time ago. Just because perhaps when the protein governs gene activity, it takes some time to produce the protein. And then, of course, when if there was no delay, normally you have a stationary point here when the synthesis equals degradation. But then assume that you see the blue segments here. It means that we have to consider degradation now here, but synthesis corresponding to the protein level here. So in this case, you see that you have a low protein level, which means low degradation, but you have also, but in the past, at time t plus minus tau, you have a high protein level, which means also a low synthesis. And actually, to understand this diagram, you have just to see that so you see that you have a circle, so it's going down, and at this point, p, the time derivative of p is 0, and p increases again, is because here, at current concentration, the degradation is given by the red curve, but at time t minus tau here, the synthesis is given by this protein concentration, which is here. And actually, the system goes up again because degradation here equals synthesis here. And so you see that in this way, because of the time delay, you are continuously overshooting the fixed point here, and you never reach equilibrium. And so in a sense, and which means that actually introducing a time delay in a cellular system or any system actually is a very simple way to destabilize a fixed point and to induce oscillations. Of course, so how do you create time delays? Well, a very simple way is just to force some additional process. So again, if you consider a feedback loop with a single gene, because RNA might be translated in the cytoplasm and protein must be translocated back to the nucleus, you have some transport. This could create a delay. So in some models, you realize that when you write the simple model, you have no oscillations. If you just add this additional step corresponding to transport between two compartments, then again, you can get oscillations. Another process which is often overlooked, and this is why the remark about the null line will be very useful, is just to have nonlinear degradation, or basically, which means protein degradation inhibited by the protein itself. So again, you see, think about cooperative stability. When you have a lot of protein, it has a way just to block its degradation. So this is what is represented here. And in this case, so this null line, which was monotonous here, just gets a S shape, because when you have a small amount of protein, you degrade a lot, but the more protein you have, the less you degrade until eventually it goes up again. And then this corresponds to the S shape of the red curve here. And then you see that now it's very easy to have oscillations. Why? Because remember that in each of the 4 quadrants, you go in some direction. So here you go to the right and up, to the right and down, and so on. And now when you arrive here, then you must go up, and it is only here that you have the right to go down, but it's too late. You do not have time to go back to the fixed point, and you overshoot the fixed point until here. And then you do this circle again. And because you force the system to go far away from the fixed point, you can never reach equilibrium. In a sense, so again, it's a nice example because you see that by tuning the nonlinearity of the degradation, then you change the geometry of these null lines. You prevent the system from relaxing to the fixed point, and then you can trigger oscillations. And then it's interesting because if you remember how we did find ways to introduce nonlinearities in the equation, then you see how you can really make a series of logical steps from the structure of the network, the kinetic equations, to the dynamical behavior. Okay. So, well, just a classical example, which was proposed as one model of the biological clock some years ago. And again, again, the same ingredient, the p-null-Klein, which corresponds actually to the degradation function, to the degradation kinetics, also has a S shape, and you just observe this limit cycle behavior. So I talked about the cell cycle. And it's interesting because I mentioned the use of a positive feedback and then a negative feedback. And in the cell cycle, you have both feedbacks cooperating to generate robust oscillations. And then it turns out that there is this positive feedback of the cyclin B CDK1 complex over itself. You remember when you just increase the amount of cyclin B at some point, it jumps to a high activity state. But you have also a negative feedback, which is that when this complex is active, it will trigger the transcription of the APC complex, and the APC complex will induce the degradation of the cyclin B CDK1 complex. Okay, so it's like a timer. When CDK1 becomes active, it creates what will degrade it. And then it turns out that, again, you see this positive feedback loop here for the CDK1, involving CDK1 gives here a red null-Klein here. And then the negative feedback loop, and so combined with a negative feedback loop, then this will create oscillations. And then this is what will drive your system around the cell cycle progression. So of course, the cell cycle is not really an oscillator. It is an oscillator during embryogenesis, because during embryogenesis, you make rounds of divisions one after the other. So in this case, it's really a limit cycle oscillator. But then, in the general case, you only make the cell division when you need to, and then this is where you have checkpoints and system to block this oscillator. But the cell cycle system can be an oscillator. Okay, so here it's another way to see it. You see, basically it's here cyclin B and cyclin B CDK1 activity. So you just evolve in one sense. You switch on, then this begins to trigger the degradation through APC complex, and then you come down, and then you just make circle around this Bistability cycle. So basically, it's the same mechanism as in the van der Pol oscillator, which was derived from the dynamics of electronic tube in the 20s in last century. Okay. So I'm trying. So again, well, just a few slides. I will briefly go over. Well, this was just to mention. Of course, you can make simple models of the cell cycle where you are just cyclin B, CDK1, and APC, just to understand this interlocking of positive and feedback loops, and how this creates the oscillations, and how this creates the irreversibility. Of course, you can make the model more complex, write equations, and then just to see what are the different behaviors of this model depending, for example, on cell mass, and just to, well, of course, it's very too brief and there is no way I can really explain all these complicated diagrams. But the idea is that by looking at all the possible behaviors that emerge from the system as a function of some critical parameters like cell mass, then you can understand really what is going on during all the biological events. And for example, here you are in some state, but then as that cell mass increases, this state disappears. You switch to another state, which is where you replicate DNA. And then as cell mass further increases, then you reach a region where you have an oscillating state. And this corresponds to the state where you have this mitosis phenomena, and then you make one turn, you make this turn around the bistability cycle, and so on and so on. Okay. Okay, so, well, I think I don't have time. So I wanted to do some mathematics, but, well, just to see how you would take a system. So this was an example of a gene with some memory, which means that the gene activity does not react instantaneously to the protein concentration, but after some time constant. And of course, I will not have time just to go over all this, but perhaps I can say that this is just say that this, well, something like that you might have encountered in other systems. It's when you trigger oscillations. I mean, a very classical phenomenon is called a half bifurcation. It's when you have a system, a fixed point, you linearize the dynamics, and so you have eigenvalues. And then when you have a couple of complex eigenvalues whose real part becomes positive, then it is where you induce the oscillations. And then well, there is a whole theory, which is absolutely not related to biology, but general theory of how you analyze such bifurcations. And just if you take this system for a self-regulated gene, then you can just find the fixed point, you linearize the equation, you have a matrix, you compute the eigenvalues. And it turns out that there are some techniques just to perhaps without having to find the explicit form of the eigenvalues, just to find when there is a bifurcation. So there will be, you can know that there will be a half bifurcation leading to oscillatory behavior, where there is some conditions between the characteristic polynomials. And then I switch to the results. So in this system, you find that the condition for the oscillations, you find a very simple condition for the oscillation, which is this one, and which depends actually on two parameters, which is t and sigma. And t is a time delay in your system, and sigma is the saturation of the degradation, which means that you have sigma around 1. It's not very saturated. When sigma equals 0, it's very saturated. Negative sigma corresponds to the negative slope that we saw. And then in this case, we see that indeed, when there is a small time delay, we really need a nonlinear degradation. And when we increase the delay, then we can resort to less nonlinear degradation. And well, it's a nice example where you can really show how you need to have saturated degradation or you need to have a time delay, but you can see also how you can trade off one for the other. And then this is a kind of analysis which is useful just to understand how exactly you can trigger oscillations in a cellular system. Okay. Yes. Another example of oscillations, biological clocks. So probably it was not so easy to wake up this morning, perhaps because your biological clock was not ready. And so perhaps you know that inside your bodies and inside each of your organs, you have a clock that is running with a period of about 24 hours, and that triggers, that really governs your physiology and adapts it to the day-night cycle. And well, it is known that it is really clocks that are running at the cell level. So it's a famous experiment where people use reporters just to follow the clock dynamics in single cells. So you see here in this picture, each colored trace corresponds to the oscillation of some gene activity in a single cell. And it's running with a period of 24 hours, with a high precision. And this to give an idea of how high this precision is, here is an experiment where they looked at cell cultures, and they looked at the average oscillation in the cell cultures in cells put under constant conditions. So usually, you know that if you have people running around the stadium, you know that after some point, they will spread across the entire stadium, just because they are running at different speeds. And again, if you have a set of clocks which are running at slightly different paces, then after some time they will be desynchronized, and you will lose the average oscillations. But you see here that it takes 40 days to dampen a little the oscillations. and which means that they could estimate that just to be off by half a day, it would take 100 days, which means that you have here a clock which is precise at the level of a few minutes per day. So one of the goals of analysis, of dynamical system analysis, is just to understand how this is possible. Okay. And then perhaps 5 minutes or so, no, 2, okay, okay. And then, so just this will be the last slide. Fortunately, it's a summary slide. The last very interesting use of oscillatory or pulsatile dynamics is in cellular signaling, but unfortunately, the next speaker will speak a lot, I guess, about the NF capable systems. Again, and then there are a few also small networks where you have a pulsatile dynamics generating pulses, often in nuclear localization. And then it seems that the frequency between the time interval between the pulses gives some encodes some biological information. And there is a lot of papers of interest in understanding how you encode biological information in such signals instead of in the constant level for protein. So thank you for attention.